Good evening and welcome,
Hello.
everyone. Hello, it's good to see you all. Thank you for being here tonight. We are excited to welcome you to our LAM Research Virtual Conference presentation on leveraging the LAM Cell Atlas for scientific discovery. My name is Cindy Beasley. I am the Director of Education and Advocacy for the LAM Foundation. And tonight it is my pleasure to introduce to you our speaker and host for the evening, Dr. Yan Zhu. Dr. Zhu is professor at the University of Cincinnati College of Medicine and Cincinnati Children's Hospital Medical Center, where she also serves as the director of the Bioinformatics Core in the Perinatal Institute. She has led the bioinformatics team at the CCHMC Research Center of the Lung Map Consortium for over 12 years. Dr. Zhu's research focuses on the development and application of bioinformatics and systems biology approaches to uncover novel mechanisms embedded in large-scale data sets. Using single-cell multi-omics and spatial transcriptomics, her work addresses lung diseases including LAM, IPF, and CF. Dr. Zhu's lab has developed widely used analytic and visualization tools such as Sincera, Slice, LungGens, LGEA, Lung at a Glance, LungMap, CellRef, and the LAM Cell Atlas. She has published 125 peer-reviewed articles and serves as a reviewer or editor for several reputable scientific journals. Dr. Zhu, thank you for your willingness to share your knowledge and your expertise on the LAM Cell Atlas tool with us. I will now turn the evening over to you.
Thank you very much, Cindy, for the kind introduction. And I like start with thanks for the meeting organizer, Cindy, Jan, and Vera, for their efforts to putting this together and give us the opportunity to present here. And I also like thanks for everyone attend this evening's events because I know how busy your time. And what I can do is try to make this section practical and useful and worth your time. So I will begin with a brief overview and introduction of the LAM Cell Atlas history and what the new features and functions we have been working on. And a little bit later, Andrew Wagner, he is our current web developer. He will join me to give you a live demo on the newest the Atlas functions in the Shiny Cell version 2. And so let's start. So what is the cell atlas in general? So atlas is built with the single cell omics as a foundation. So it's provide a single cell resolution map to all the cell types, states, and interactions. So it's commonly used as a reference framework to integrate your own studies, such as multi-omics and spatial, and even the clinical data. You can use it as annotate the metadata. And it offers a user-friendly interface, which allow the research without coding skills to explore and reanalyze their single cell data. It promotes the collaboration by connecting the clinicians, biologists, and computer scientists around the shared data source, and it's increased the accessibility and the reproducibility. And with the ultimate goal is to accelerate the therapeutic discovery. On the right panel, I put some of the examples on the LAM Cell Atlas. And for example, Minze and I put together the LAM map cell ref for both normal human and mouse LAM. And then at the same time, Human Cell Atlas and HubMap worked on their own version of the normal and the disease LAM reference. And we also closely work with them through the cross consortia kind of the work together. And the first disease LAM Atlas map was released by Kaminsky's lab in 2020, which is the IPF cell atlas. And they also have a COPD cell atlas. So the COVID cell atlas released after pandemic in 2022. And with the support of the LAM Foundation and the close collaboration among 3 institutions like the UC, CCHMC, and the BWH and the UPenn, we put together the LAM Cell Atlas released in 2023. And last year, the end of last year, and there's a Union LAM reference released. It's collected more than 9 million cells and 1,800 donors across 17 lung disease conditions. So it's getting more and more sophisticated. So what is the LAM Cell Atlas? We designed it as a centralized interactive platform to integrate all the single-cell omics data from the LAM samples, excuse me. So it's built to leverage the recent advance in the single-cell application in LAM. And who is this for? And in my mind, we designed for the LAM investigator without coding and bioinformatics experience. And for clinicians, scientists, translational research, and web bench scientists, and they can quickly explore the gene expression, cell state, and ligand receptor communication, for example. And this can use for cross collaboration, and so because it's a robust data resource to test a new hypothesis. And this is not designed actually for computational power users, because we tend to use the raw data for customized analysis. So LAM Cell Atlas is like I mentioned, created with the strong support from the LAM Foundation. We emphasize the community share goal and to accelerate the discovery and improve the outcome for the LAM patients. It makes the single cell resource open, reusable, and facilitate the identification of like novel biomarkers, pathologic pathways, and potential therapeutic targets. And those are evidenced by the recent publications. So in these slides, I summarized the LAM-related single-cell publication into 3 phases. And so for phase 1 is more in the discovery, looking into the LAM cell type, identification, and its origin. And phase 2 is more getting into the mechanistic study. And phase 3 is has a trend to looking into the translational and emerging of the spatial technology. But the phase is not like, it's an arbitrary cutoff. It's not a clear separation. It's not means like phase 1 paper don't have mechanism and phase 2 don't have the translational study. And in fact, it has a lot of overlap just by looking the name and the paper. And the clear trend here actually is the cell number increase with time, and the technology, new evolving technology come out. Just using our own data as example, for 2022, the paper, we used 22K of the LAM cells from 4 LAM patient. The technology is the single cell RNA and the single nuclear RNA. For this year, the new integration, we have 650K of cell from 20 patients. And the LAM core alone, we already have 50K. That's allow us to have the power to look into the subtypes. And the technology, you can see it's we have RNA attack, multi spatial, and so it's this tells us the Atlas is a dynamic resource. It's evolving, grow with the technology and with the data type. And so this leads us to the overview of the LAM Cell Atlas web architecture. The blue box are the current version 1 that you guys are using. So at the front end, you're sitting in front of your computer, and we designed mostly using HTML and JavaScript. And you have a query, it's sent to the backend, the servlet, and the servlet talk with the Oracle database and retrieve your results. And the current Java setting is good for the exploratory kind of the research, and but it's a lack of sophisticated analytic power. So, for example, if you wanted to do on-demand interactive plot, it's hard to achieve that. Another limitation is the current setting, the Java environment usually cannot process over 30K of the cell numbers. But for the new study, it's for any of the spatial study, it's easily over 70K of cells. So for that reason, we launched the version 2. And so we added the backend, basically connect with the either R Shining or Python in the backend. So it's improved the analytic capacity. And then for the database, in addition to expand the data sets, current data sets, we also add the new the data modalities such as the spatial. So this is an overview of the data summary. The red color shows the version 2, and you see we put the spatial data and also some of the gene regulator network data. This is the LAM Cell Atlas website from 2025, and the top 5 cities are Cincinnati, Nashville, Nottingham, and Rochester, and Boston. And the top 5 countries are United States, UK, China, Ireland, and Canada. So I just want to remind you that all those stats come from the production server. So like Andrew and I working on the development server is not contribute to the traffic. Next, I will give a live demo. If you have any question now, or we can wait to the QA section. And so the live demo, I will consider some of you are new for the Atlas. I will walk you through like the main web interface, the major functions, and how you found the right tool, right data sets for your research, and how to connect using some of the interactive features such as LGA, Top Fun to deeper your analysis. And later, Andrew will jump in and show the Shiny Cell version 2. And I think this is a big step towards like the intuitive and user-friendly plotting. And I think you would like to see what it can do. So this is our webpage. And so we have the LAM Foundation mission, and we have the accumulated data points. And we have the top row is already in the version 1, which is transcriptome, epigenome, connectome, and the project, except this year we're adding new published single cell project, such as our niche paper and Sammy Johnson's the Sinensis paper, and also I think that's from Vera's group, the 3D surface model and the spatial transcriptome, etc. So you're welcome to contact with me, and if you have a new publication related with LAM single cell, we're happy to add in. And this is showing the funding. It's funded by LAM Foundation. And for the new person, new user, I think I show you the about page. And this is the version 1, the function, and you can read and have some sense what it's doing, the query by gene, query by cell. And actually, right. So the new things here are listed under LAM network and spatial transcriptome and the shiny cell, which me and Angel will go through. I will go through some of my examples. I built some user case. I think the most common usage are, people has interesting gene in their mind uh, for a biomarker, for example, analysis. And I will use the, the MDK as example. But you can have any other things in your mind to do the same thing. I just show you here. So you can choose a gene query and you do the MDK. And give a minute. Okay, so we give you the UMAP and give you the bar graph, and it's abundantly in LAM cell, but also in other cells such as goblet cell, etc. And you can see the expression and some of the statistic, the enrichment and t-test. And you also see, we often get being asked, so what about the protein expression, right? So you can link either to the external link to the protein atlas. For example, we have a mouse human. If you're interested in, say, the mouse development for this gene, for example, you can see, oh, actually the protein RNA is quite correlated for this gene through the development. And let's see if you want to see human protein atlas, and that's give you much more, very enriched information. And you can see the protein expression across different tissues, and you see, okay, it's enriched in the endocrine and GI, and it's really highly in the female tissues. And it's clustered together with the ovary. You see, if I'm interested, what about those gene expression in cancer? So you can click here. You can see, okay, it's highest in the uterine endometrium carcinoma, and it's also very high in the ovary cancer. It's generally high in all the cancers. Here is the lung cancer. You can see it's significantly high. And so basically you can find a bunch of additional information. If your project is built on MDK, you can have a global view of this. And the second user case I would like to show is, remember, so in the LAM symposium, Dr. Lisa Hansky asked me a question about the difference between the LAM and ML. In real case, this is actually a big project. You needed to basically collect enough sample from both disease. And for Cincinnati, for example, we do not have many ML single cell. The things in the database are mostly contributed by BWH. So basically, we probably needed the collaboration to put all those data together, and through a properly integration and a comprehensive analysis to tease apart the what is the common and what is the difference part. But today, I just want to show you using what you can do in LCA. It can give you a quick feasibility evaluation, and it can give you some preliminary data, generate some preliminary. Okay, so let's do that. Do a comparison. So we do the LAM, so we can see the LAM call signature. and you can click the LAM call signature, and you can, of course, search one by one, and we also give you a lot of external link, for example. But so for all those links, you can search for the gene you're interested. But mostly, we can just download the table, and you also can click this, and they show you the enriched function of your gene list. And for the sake of time, I already downloaded, and then after that, you say, okay, I got the lung signature. What about kidney? So you pick the kidney, and then you select Ian Tan's publication, and you can see, okay, I want to query the cell type, and I want to select, for example, tumor cell type from their study. And then you download the table here. And so here they have the top 100 you can download. So after download, you can compare. You can compare using many tools, but you can using our in LGA is another the web portal we developed, and so you can we link on the LAM Cell Atlas homepage. You can just click and go there, and then so you say, okay, I want using the signature comparison, and I want to use a user list here. And so you downloaded the two list, right? So I have the LAM call from here, you copy and you paste. And then you go back to put the ML signature. You copy and you come back and you paste here. Okay, and then you can show okay, so for the top 100, you get the 86, 87 overlap. That's a lot. So you can look into the unique versus the common ones. So you can download, and of course, you can also copy this. For example, I have another list. If you want to say, okay, what are the transcriptional factors in this list? What are the surface markers are in this list? So I can use for my research. So here we have the tool. You can extract transcriptional factors or surface markers, and you can just, for example, paste your list here. And you can run so we can extract the things from here. And I already extracted. Here you can see in this list, you have the common transcriptional factor like estrogen receptor 1, HOXA10, and PGR, and SNAIL2, and TCF21. Those are common from these two diseases. And oops. I just want to show you, but it's okay, let me do it again. It's excellently wiped. Okay, so you have the 87 common, and you can see, for example, you can check the cell type distribution of those of those genes. You can also see enrich the function of those genes, and so since it's only 87, we say we don't want the family arrow control, and I just start. Okay, we see those common ones are mostly like extracellular matrix proteins. Um, yeah. What about the pathway? We see the extracellular matrix, and I see a big defective collagen degradation. Syncam pathway and ECM and neurogenesis. And I also see a big integrin pathway, and I see a big EMT somewhere. But anyway, you get my point. So you can do the what are the common markers, what are the common functions, you also on the pathways, and what is the potential upstream regulator for those. So those can give you some of the preliminary data to evaluate the feasibility, whether you wanted to launch a big study like that. And I think with the giving the time limit, I wanted to. I know many of you interested into the spatial, and let's look into the spatial. And so this is still under development. Currently, we have 4 of the Bayesian data and 2 of the Zini data. We just put in to give the try to build the map. And those are also funded by LAM Foundation to Dr. Minze Guo for his career development fund. He used that for the biomarker study, and we use it for the niche paper and also for the LAM Cell Atlas to start to building the spatial map. And Dr. Jian Yu from UC provided the sample, also did the lesion selection for the spatial transcriptomics. So you can select any of those and explore in the cell time gene. I give you, so this is the Vision 1, for example, and Vision version 1 is not in the single cell resolution, it's spots level, but you're still able to see like a so for example, you can highlight, you can, this is color by cell type, and you can annotate the cell type. This is the LAM one, and this is lymphatic endo, and you can, for example, put the PML, and you can color by PML. See, it's mostly in the LAM call, and you can see the LYVE one as a lymphatic endo marker, and if you color by that, it's in the lymphatic endo. So it's overlapped, so each spot may have multiple cells, but you still can see the gradient. And for example, you're interested in the ligand receptor, you said, okay, VEGFD, and it's mostly in the LAM call. And for the receptor, you for the FLT4, and it's mostly in the lymphatic. So this is mediated the LAMCO and the lymphatic. And the Xenia has a much higher resolution, and it's a single cell, even the subcellular resolution. And I have this. This is a Xenia sample, and you can again color by cell type, and you can label the cell type, and you can see the gradient, the separation is much better. You can identify more than, I think it's 29 cell type. And so the LAM call is here, lymphatic endo is here, LAM call 2 is here. We have some of the F1, F2, and the capillary. So I said I want to show you how to calculate using, so some of your user probably not satisfied just by a single gene alone. And they want to show a pathway, a list of genes that are interest. So I can give you example how to put a list there and calculate the module score. And I have some examples here. So for example, you're interested in the ECM and the collagen genes, and you can copy and putting is that I want to create a new gene sets, and you can put ECM. And so here I created a module, and you know the Zini is not whole genome, so some of the gene are not there, which is fine. You still have a good panel to represent the ECM. And if you see, okay, the highest is in the middle here. It's the LAM-associated fibers, F1, F2, and the LAM-CoL2 also has the a good gradient there. And for example, we can have another example if we interested in the synthesis genes, and we can copy the whole thing. And we can create a gene list. From Mayo Clinic. So, and then you can see, okay, it's actually pretty high in the endophilia and the epiphilia, like 81, 82 here. If you don't remember the cell, you can just go back and you can color again by cell type. Now you can see this is 81, 82, this capillary endophilia, they have a higher synthesis, for example, activity. The other thing I want to show is so this can do some of the statistic, like the t-test. For example, you let's see, if you put this open the cell type, you can hover over and see each cell type. And for example, if I look into the capillary, I found two population. And one is, can you guys see, there's the kind of the light orange color. There's one here and also one here. If you put in here, you can see. So I said, okay, what are those? Are this different? These two capillary endophilia are different or not? And they're spatially separated. So we can just select, for example, capillary, and we can just show this two subtype. And we like to see the difference. We can select also this part, and then we can count as cell 1. And then we select this part. We can put into cell 2. And and then say, okay, so what are the difference? So now it's show you the difference. What is higher in the cell 1? You have the 17R, IL-17R, and then for the version 2, so you can get the markers, and so you have the ED and RB, and I will more familiar of those markers than you, so I can say, okay, this maybe is actually one is the cap1, the G cap, one is the cap2, which is the A cap. But if you say, I don't know those markers, and we have the tool for you. So if you go to the, if you go to the LAM Cell Atlas, you just click the LGEA and using our LAMAP single cell reference. And So basically, if it takes some time to load. I think I have one open here. So we just, instead of waiting, we can see here. So uh, this is selected for MDK. You can, of course, you can search for MDK for the normal expression here. Uh, but let's deselect. So when we deselect, we have everything, right? So you can click here for CAP1 and CAP2, and you can search for markers. For example, you see the IL7R. So okay, so this is a CAP1 marker, and you see the EDNRB. This is CAP2 marker. So that two capillary in the spatial, as maybe it's a cap and a G cap. So I think I will give the time to Andrew because he has some exciting new tools to show as well. And you guys also feel free to ask me questions, and I can check my chat box now once I finish the talk. So I will stop, and Andrew, you can pick up.
Okay, great. So I'm Andrew Wagner. I've been working with the lab as a web developer, developing a new version of Shiny Cell app. So, like you could just see, you just go down to the section for the Shiny Cell app. It'll take you here. This is the home page. This app was developed with an SRAT object that held data, RNA data from both control samples and LAM samples. So,
Yeah, so this is a new feature that users requested because the previous version we don't have controls, and this is the new integration that is
mhm.
still, under revision of our paper. But so we will release a complete version with control and LAM.
Yeah. So the app has three basic components on this page. We have our plotting instructions on the left, the main plot in the center, and then a data table that's meant to split continuous data into user-defined bins. That'll make more sense in a moment. But just scrolling down, we can look at the central plot. Right now, we have cell type selected, so all of the cells within this dimensional reduction are going to be colored by the cell annotation. So from here, we can just scroll down. One of the new features in this version of Shiny Cell is that we can click and drag to look at a specific section of the plot. So we can see that there's a lamp core population right here, clustering between smooth muscle cell and fibroblast. So we can just click anywhere to undo that, and we can look at we can start to look at more continuous data. So if we just go to this drop down, we can select RNA from the plot, and this will just plot any kind of expression data that was within the object. And then for the sake of clarity, we go down here and we can play with the plotting options. So switch the scale from previous one to white to red and plot, change the plotting order. If we scroll back up, we can see that PMEL, which we've been using as a LAM core marker, is selectively expressed within the LAM core population that we just looked at. Just even when you scale it, you zoom in, you can see that it's definitively within that LAM core population. And then on the right, you can see that the data table now uh, it treats 4 as a default, but it can be set to whatever you want. So if you wanted to do 10, it'll automatically split it into 10 bins, and it'll also calculate the number of cells within those bins, and then the number of cells in the viewing window for the zoomed in section. For our plotting purposes, this is great, but it's kind of difficult to look at the expression of a specific gene and then also compare it to the cell annotation. So that leads nicely into our next feature. So in this feature, it allows you to plot two-dimensional reductions right next to each other, and you can choose between any of the previous options for what you want to do. So in this case, on the left, we have the cell metadata, we have the annotation for each cell type, and then we have the expression of our marker. So you can see that it's just nice to have both side by side. And then if you scroll down, there are additional panels that give you a better view of the comparison. So the left panel will come into play in a moment. On the right, we have a table that just shows you the number of cells within each cell type label, and then the number of those that are actually expressing the marker. So you can also sort this list, and it's very clear that you can see that 50% of the LAM-Core 1 and 50% of the LAM-Core 2 cell populations are expressing this marker. And then the next one down is goblet cells with 1% of them expressing this marker. So from here, and just for the sake of looking at we can change the metadata we're plotting to conditions. So this just shows, like we said earlier, it's an integrated object with cells from control donors and LAM donors. This can just show you where they place on this dimensional reduction. You can see that the LAM core population is unique to the LAM samples, as you'd expect. Now we can go in. So this is just a comparison between plotting the metadata and the expression. We can plot metadata to metadata. So if we just select cell information here. So we have the disease condition on the left and the cell annotation on the right. If we scroll down, the left panel is now changed. And it just shows you a heat map breaking down the number of cells within each of the shared groups. So as you can see here, there's 0 LAM core within the control. Then just a quick overview of the distribution of cells within the cell annotation and split by condition. The table on the right just shows you the same data in the form of a table. And then all the data tables have the same functionality. At the bottom, you can copy to your clipboard, you can download it as a CSV or an Excel. All of the data held within. So from here, we're going to look at expression compared to expression, and I have that open already just because this is by far the step that takes the longest, is just loading the expression onto the plot. So in this case, we're looking at the expression of PMEL on the right and the expression of CTSK on the left. So you can see pretty clearly that they're most strongly expressed within that LAM core population, with CTSK having some expression in the fibroblast cluster below. So in this case, the panels below this have changed. On the left, it shows you a plot comparing the expression of the two. And then on the right, there's a data table that holds several correlation metrics you can look at if you want a more quantified analysis of the co-expression of these two genes. Now, if you are interested in having a co-expression plot where you look at the expression on one singular dimensional reduction, the app also has that functionality. So we just go to the top, we click on RNA-seq, and then we go down to gene co-expression. And you can see that you have one singular dimensional reduction with two-dimensional legend here showing the co-expression of the two genes plotted over the original UMAP. And then we also have a table showing the cell numbers and the percent of each for the expression of these genes. So from here we can look at the next feature. I'm sorry, that's a holdover from when I walked through this earlier. So let me just start from the very beginning. So we load the violin plot feature for the app. We come down here, we select from the options what we want to plot on the y-axis, and it's already set to cell type on the x-axis, so we'll keep it as it is. The default is PML for a lot of these. You can see very clearly that PML is still specific to the LAM core. So in this case, as a demonstration, say we wanted to look at a different type of marker to see what its expression looked like. Or before that, let's see if we wanted to look at a box plot instead of a violin plot. We can toggle between the two, just like that, just by clicking these radio buttons. So we can just type in whatever marker we want here. I'm going to use ACTA2 because it's a smooth muscle cell marker. And you can see that it plots it very quickly. You can look at the box plot here. You have everything you need immediately. You can download this plot. You can set the height and width, and then you could be fine here. So at first glance, you can see that there's close expression between this and LAM core 1. First, we would want to make sure that we subset this to just look at LAM core samples. So you can click this button here, the toggle to subset cells, and we can select the condition option from the dropdown. So once we've done that, we can just unclick control. And now the plot is only plotting data from cells from LAM donors. Once we've done that, we can still see that LAM core 1 is expressing ACTA2 similarly to the smooth muscle cell. So we can run a statistical test here. We just click on toggle to perform the statistical test. And as a first step, we can run a global test just to see if the variation between these groups is greater than the variation within each sample. So we can just run a quick ANOVA. And we can see that that is the case, that there is a statistically significant difference in expression between groups. So we turn that off. Now we can look specifically at LAM core 1 and LAM core 2 compared to smooth muscle cell. So we just scroll down and we can perform pairwise tests. So if we select Wilcox.test, we can run a Wilcoxon test on these 3 categories. So we just go down to this input box here, and we can type in the pairs separated by a comma to perform these tests. So we compare LAM core 1 to smooth muscle cell and LAM core 2 to smooth muscle cell. And from this, we'll get a p-value for each test. And in this case, you can see that while there's a significant difference in expression between LAM core 2 and smooth muscle cell, there is not one between LAM core 1 and smooth muscle cell. So they're both expressing this marker in a similar way. From here, we can go on to the next feature. In this case, this is a proportion plot. And this one is very straightforward. It just shows you a breakdown of each category on the x-axis. So we want to look at the distribution of cell types within the healthy control versus the LAM samples. This is a very informative way to do it. And then you can download this plot. You can subset the cells similar to the previous example. The only difference is that you can also toggle this to look at just pure cell number. Okay. From here, we go into the final feature for the RNA data. So this is a tool that allows you to plot any kind of heat map or bubble plot for an input list of genes. So in this case, It's set to bubble plot. And before I go any further, let me play with the graphic controls for the sake of clarity. So we're going to make a large plot with a small font just to make it legible. You can also see right now that the donor IDs are along the x-axis. We can change that by going to group by and selecting cell type. So the default inputs are cell-specific markers we use to identify cell types during the analysis. We can just, as long as your list has 50 genes or less, you can put in whatever you want. So in this case, we're going to use genes associated with the extracellular matrix. So we just input those. You can have a plot like this. Now, as a default, the app will plot, it will cluster the rows. That's a feature you can turn off just in this section here. You can toggle scaling and clustering based on whatever your needs are. So we're interested in how it might cluster the columns. So, and we're going to set this to heat map. From here, you can see it's a bit difficult to get both in, but you can kind of see that the clustering shows that it breaks down along lineage types. So all of the endothelial are clustered together, all of the epithelial are clustered together, and all of the mesenchymal are clustered together, with the highest expression being in the fibroblast cell types. So from here, it has all the same options as the previous one. You can download the plot, you can set the dimensions, you can subset cells, and you can play with the graphics. However, whatever would be the best for your use. So that covers all of the functions available for the RNA. We also have a tool for the attack. So I have that open already. A lot of the attack is very similar to the RNA. So I'm only going to cover the parts that are unique for this. The only difference is that the attack, it has all the functionality of the RNA, but it's done on gene activities inferred from the epigenetic data. And it has motif activity scores, again, inferred from the epigenetic data. So back to the plot. This track plot is similar to the genome browser plot. You can just look at all of your data. So this comes from another Seurat object in our most recent paper that's currently being reviewed. This object only includes cells from LAM patients, but it's an integrated object holding all that data. So it gives you the option to query either a region or a gene. So we're focused on genes, and we're going to select a gene that we know is highly expressed within the LAM core. So you just put your gene in. Now this one differs a little bit because you do have to click the update view button for it to actually plot a new one. And for the sake of visibility, we will also show that you can play with the height and width of the plot. Oh, wait. So here we have a more condensed plot showing that EMX2, the LAM core track, has much higher expression than all of the other cell types. And then it also shows the genes below. Finally, so Like I said, it has all the same functionality as the RNA app. This is our dimensional reduction from this object. With epigenetic data, we had a lot of issues with hitting the same resolution as the RNA just due to technical factors. So in this case, we have much more general annotation, but all the same functionality. So in this case, you can plot the metadata, or you can go down and you can select ChromeVar, which will be predicted motif activities based on that epigenetic data. So we added a panel down here where you can actually visualize the motif sequence that you're for the transcription factor that you're visualizing. And like all of the other plots, it has the ability to download it in whatever format and with whatever dimensions you want to use. So that covers all of the new, that covers everything new for the Shiny Cell app. Let me just stop sharing my screen.
Okay, great. So I would like to give the acknowledgement to the people contributed to the LAM Cell Atlas development. In my lab, and Ina Du was the previous developer for the version 1, and Andrew Wagner is here. He's the current web developer, and everyone here contributed on the data analysis and integration. And Dr. Minze Guo, now he has his own lab, but we're still working close together as a team and share resources, share ideas, etc. And I like to give thanks to my UC collaborators, Jan Yu and her lab, Dr. Frank McCormack, Nashant Gupta, and Cope. And I already mentioned for the LAM Cell Atlas version 1, we have close collaboration and the data sharing with BWH, with UPenn. And so this is community efforts to put together. And I also have close collaboration with Steve and Simon Johnson. And I'm glad to see their lab is actively using the resource from the web stats. And for the CCHMC, I like to give thanks to our strong leadership support and Dr. Wisset, Dr. Ji Chen, and my LAM project collaborator, Dr. Catherine Weckenhanser Broadcamp, and Anna Purr, and Deborah Schinner. And I also like to thanks for the DCC, especially for Mahir, helped us to set up the R Shining server. And I definitely like to thanks for the LAMS Foundation and Jan, Sandy, Farah, we being talked back and forth to set this event. So I really appreciate all the efforts behind the scenes. And last but not least, I like to give thanks to the LAM patients and the family and for their generosity and partnership to make this work possible. And thank you all to spend your evening here. And I think I'm open for any further questions and discussions, and like to hear your feedback and thoughts.
I have a quick question. I noticed your LAM cell, no, the control cell has some LAM core 1 and a little bit of LAM core 2. Small population. Is that correct?
It's very small in a single cell. I think it's because the technical, the cell isolation, we lost a lot of those. But for spatial, they're very abundant. Yeah, you get thousands of more than,
Even in the control samples?
Control, we have 0. We don't have, LAM cell.
No, but I saw with Andrew's presentation, there was a very small fraction of LAM core cells that were present in the control.
It's probably because the color is overlaid, but he has the table under, okay. And if you pay attention to the table, it's 0 LAM core in the control. Yeah.
So I also know it's still under development. Do you guys have an idea of what time frame it will be ready?
Mm, I think the shiny cell for the transcriptome should be released soon. Yeah, we're testing. We're also waiting for the paper because the data is all from the new integrated paper. As soon as we heard anything from the revision, and we will release. Yeah.
Thank you so much, Dr. Sue.
Mm-hmm. Yeah, we will gradually release. I think a transcriptome is ready, and then after that will be the epigenetic. We still try to adding additional features like the chrome bar, all those, and the motif are newly added, and we can add some statistic whether how the motif whether is enriched in some regions, and I think spatial. Angel is currently trying to. I only can show you static like the lens tissue and the UMAP, but what we try to do is overlay the UMAP on the tissue so you can see the real niche in their native environment. And we have some alignment issue. We still need working on that.
Yeah,
Has there
that's the
been,
problem. Sorry, go ahead.
real quickly, has there been a way to correlate your epigenetics data with, like, see if there's some kind of overlay between your ATAC and your RNA data to see if there is some compound mapping that can go on where we could relate epigenetic mechanisms with just um, maybe if they could influence transcription factors or their expression, and maybe that connection. I'm just thinking if you guys have thought.
Yeah. Yeah, I agree. I mean, I like this approach, right? So we give you the resource, and actually, you do need to put your own, like a design and idea to use that. And but this is a good point. So basically, you want to see the upstream, and I tried a little bit of showing, but not too deep. So the best way is actually the gene regulatory network, right? So once you have the network, and you can identify the nodes, and so hopefully the hub will be the important transcription factors, and you can follow
Yes.
your hub, found all the target genes. Yeah. This is and in the still early phase of the development. We have some map, but it's still a lot of work to do, and so we didn't show today, but we do have some map. I mean, if we have time, we could show, but I think I rather save some time for the discussions. But that's exactly in our plan. Yeah, and you also can through the motif enrichment to basically show the if there's some of the common upstream maybe have share the motif, right? So suggest those may be the potential targets. Other questions, suggestions, and something you like to see, and you like us to work on to help you for your research. I mean, for the whole purpose is try to see if this can be useful, and you can take home and do for your own research. Yeah.
Yeah, Yan, hi. This is Jane. Thank you so much, Yan
Hi,
and
Jen.
Andrew, for the excellent demonstration and introduction of the current and the future, or the shiny version. So my question, maybe others will have the similar question as well, is what happens if we, like in the individual scientists, would try to use what you have been you have illustrated today, and encounter some questions and the technical uncertainties. How can we interact with your team so we can get our analysis moving forward without waiting too much longer?
Yeah. So I think we in the homepage I have my contact information there, And of course, it's not for you, right? You can reach me, and but for everybody, you can find my contact info, and also under the about page. And I think both me and Angel's email information is there. Sorry, I have a little bit.
Yeah, excellent. Thank you. Even for me, I will need to to get that technical support, during my visit to the
Yeah,
website.
yeah.
And the second question is, if I use the newly developed version and the previous one, the Atlas one, and I'm ready to use the plots or whatever the plot you to the UMAP or the plot that Andrew showed or heat map, how are we going to include the effort that you put everything here? How can we cite, or we need to do the author? How do we handle the authorship or the citation?
Yeah, you cite, so for example, for the previous version, we have we said, if you use this version, please cite our version 1 paper. And if you cite, if you're using the new version, because the data came from the integrated paper. So once the paper out, we're going to put the citation there. So basically you need to cite the right citation, and so that will give you, so people can reproduce using the same data sets basically. Yeah. And if you need a deeper collaboration, so if the web version is not enough, then we'll discuss the co-authorship, all those.
Yeah, thank you. Yeah, very exciting to know the new features.
Thank you. You contribute to the new spatial. I think, let me see. Vera has some questions. I have, let's see. Can the ataxic data be used to do footprint? I think, yes, you can. For the footprint, you need the row, the peak region, right? And I think for that, we can go through either the material transfer, because it's human data. Probably easier is from the GitHub. From not GitHub. GitHub is the coding for the dbGaP. If you have an ID, and we can give you the LAMAP, a GitHub account, and you can access that the fragment data you need for the ataxic to do your footprint analysis. And I don't know. I will hope it can be easier, but it's human data. I don't want to step onto the something that give me trouble. And maybe I don't know. Maybe collaboration. Also, we can directly share data, and we need to discuss that. To be clear.
So my question is just like, let's say, there's a de-identified, we can identify samples based on like their NDRI number. Like if people are acquiring samples from NDRI and happen to do like different analyses with the same donor, is there a way to kind of take that into account or link that through like a metadata? I'm just curious, like, as the tool continues to grow and more publications are kind of lumped in.
Yeah, that's a great question. I think so for all the consortiums, there's the headache issue is that we try to collect as more detailed the metadata, so we can actually put into the any of the at least we should have a totally indexed ones, so we can reach back. Yeah. So for our lab, we currently, we're contact through, for example, through Catherine, through Jen. We try to build we at least we know where the sample, it's still de-identified, but we know, initial sample number, we can reach to that person to get more information if needed. Yeah, but this is a great idea, and we have a lot of discussions through the LAM app, through the Human Cell Atlas. They all suffer the same issue. Yeah, it's everything de-identified and put together. How you tease apart. Yeah.
Yeah, I think just thinking about it up front, it sounds like you guys are already doing that, but yeah, it could be great long term. And then we've generated some long read hi-fi sequencing data.
Wow.
And so we are working on the analysis right now. But eventually we would love to be able to contribute that data to this tool. And we can talk more offline, but just
Yeah.
kind of toss that out there and see what you thought.
Oh yeah, that's exciting because I think the field needs the long rates. I mean, a lot of people ask us about which cell has the mutation. We said, sorry, the 3' cannot rate that far. And I think we always been talk. We need a pilot to looking into the long rates 3. So you actually can see the mutation, right, for each cell.
Well, so it's bulk. It's
A
not
smoke.
it's not
Okay,
single
yeah.
cell.
So it's not gonna solve that issue, but we at least we can see like it's from TSC1, TSC2, or it's mixed, right? Something like that.
Right. And we also get like methylation data. So that may be another layer that may be added,
Mm-hmm.
additive, but that's what got me thinking about the, sample IDs, because like we've done this sequencing on, let's say 35 lung samples that we got from NDRI. And so I imagine, there's a chance that those have been used for, potentially access for some other type of analysis. So that's where it'd be cool to be able to link it together.
Uh-huh. Uh-huh. Yeah, yeah, definitely exciting. And also I will be interested if some of you actually doing the single cell long rates. Yeah, that would be really exciting because this is a field issue, right? Because I keep being asked, can you tell me which portion of your cell had the mutation? And I feel like this also discussed in the LAMAP, the LAM, international symposium a lot. Yeah, so I think somebody jump into a pilot. We're happy to analyze that.
But just, I wanted to say, like, this is an incredible amount of work and so much data that you guys are working to just make it accessible. I think it's amazing for the research community and like very collaborative. So appreciate all that you guys are doing.
Thank you for the great question, and yeah, looking forward for discuss more on your data.
Hey, Dr. Sue, are you ready to wrap this up, or you want to wait a few more minutes?
Yeah, sure. Yeah, so we can give a couple of minutes back to the free evening.
Yeah, so if anybody has a last minute question, now's the time. We'll watch for just a minute.
Mhm.
All right. Yes. Well, thank you so much for this informative session. This was quite groundbreaking for us in terms of webinars that we do, and it was wonderful to have such a great turnout and to have such an incredible tool available for our researchers. I'm in awe. So thank you so much for all that you do for LAM research. We really appreciate you and all of the researchers and those who joined us tonight. So we wish for you all a pleasant rest of your evening. We get 10 minutes back for your evening.
Yeah.
And yeah, which is great. So, all right. And then your contact information, as you said, is on your website. So if
Right.
anybody needs to reach you for further discussion, it can
Yes.
occur there. This has been recorded. We will have it up on our website so others can view this and you can access this again at another time. So thank you all. Have a wonderful evening.
Bye.
Bye now.